Well, thank you, Maddie and Claire, for that wonderful presentation. My name is Laila Johnson, and I'm an incoming senior at Northville High School.
My name is Harini Srinivas, and I'm an incoming senior at Troy High School.
So during our 8-week-long internship, we had an amazing time working under Dr. Surya studying preterm birth. Although we work closely with Dr. Condon and her mentees, Harini and I have centered our focus in studying the epitranscriptomics behind uterine quiescence, focusing mainly on mice and human pregnancies. Our presentation is called Methylation in What Station? Gestation, and we hope you enjoy it. Enjoy our baby. Oh, they're so baby. So to begin, I would like to discuss the fetal stages of mice. Unlike human pregnancies, which usually last between 37 to 42 weeks of gestation, mice pregnancies are much shorter, and they only last 19 days. Although the length of gestation differs between mice and humans, the general idea of stretch in both pregnancies are the same. So in this picture, it shows a period of 10 days of growth in a mouse fetus. The greatest periods of growth occur between day 11 and day 13. And then if you look, day 17 and 19 are probably the least growth occurrence, because this is when like the final stretching of the uterus occurs. And this stretching is due to ER stress and the unfolded protein response.
Next, we have ER stress during gestation. Since the previous group spent a lot of time covering ER stress and the unfolded protein response, we will focus more on one component of response: translation and the phosphorylation of PERK. When GRP78, also known as BIP, dissociates from PERK under ER stress, it activates the phosphorylation of PERK. This activated PERK then phosphorylates the eukaryotic initiation factor 2 alpha, leading to a general inhibition in protein translation. However, the phosphorylated EIF2 alpha also preferentially increases the translation of the transcription factor ATF4, which in turn upregulates CHOP. Failure to lessen ER stress then leads to CHOP-mediated apoptosis, also known as cell death. This process is evidenced by the Western blot you see on the slide. As we see an increase, but then a decrease in BIP expression during the gestation period, there's a decrease in CHOP expression, but an increase in phosphorylated EIF2 alpha and phosphorylated PERK expression. Next, let's take a look at CAP-dependent and CAP-independent translation. CAP-dependent translation begins with the eukaryotic factor 2 binding to the 43S ribonuclear protein complex. Then the EIF2 43S complex binds to the 4E, 4G, and 4A at the 5' cap, initiating translation in a CAP-dependent manner. Lately, it has been found that in CAP-independent, also known as M6A-mediated translation, capped mRNAs don't need the EIF2 complex as they are translated during times of ER stress. The M6A mark in the 5' UTR works as a substitute for the 5' cap by recruiting the 40S complex, ultimately triggering CAP-independent translation.
Now we're going to center the discussion around M6A and its various functions. From this diagram, you can see that M6A not only plays a role in CAP-independent translation, but also transcription, alternate splicing, alternate polyadenylation, nuclear export, mRNA degradation, and mRNA stabilization. In this presentation, we will mainly be focusing on alternative splicing and translation. Let's first take a look at alternative splicing and M6A's role in it.
Very simply, when we talk about alternative splicing, we are referring to splicing a gene at its splice sites. In general, a gene is made up of both introns and exons. Alternative splicing is a process during gene expression in which certain intron sequences are moved. The final mRNA thus consists of the remaining sequences called exons, which are connected to one another during the splicing process. Now, you may wonder how this is related to hnRNPG, which is shown on the slide. Well, recently there was a paper, which is named on the slide, that discusses M6A's involvement in alternative splicing. Specifically, the paper talks about how hnRNPG binding of mRNA is enhanced by M6A marks and how hnRNPG is a known reader of M6A. In future slides, we will talk about how hnRNPG has previously been shown to be gestationally regulated. We will also further discuss hnRNPG's involvement in alternative splicing.
So now let's take a look at hnRNPG expression in pregnant human myometrium. As you can see from the image, hnRNPG is very highly upregulated in pre-non-term laboring uterine sample. However, by term labor and term non-labor, hnRNPG expression is definitely much lower.
Next, let's move on to discussing ER delta 7 and its role in regulating uterine myocyte contractile responses. First of all, ER delta 7 is one of the 3 major ER alpha splice variants that have been identified in the human myometrium. The others being ER alpha 66, which is the full-length isoform, and ER alpha 46. ER delta 7 specifically is a result of skipping exon 7 in the gene sequence. Functionally, ER delta 7 functions as a dominant negative, meaning that it blocks contractile-associated proteins like connexin 43. The image on the slide shows a collagen contraction assay. Looking at these 2 petri dish images, you can see that ER delta 7 expressing cells contract way less than ER delta 7 non-expressing cells. This demonstrates that that overexpression of ER delta 7 inhibits connexin 43 expression and disrupts the uterine myocyte contractile ability. Next, let's take a look at how hnRNPG regulates alternative splicing of ER delta 7. Just as hnRNPG is a breeder of M6A, it is also involved in the generation of ER delta 7. From the diagram, we see that if we knock down hnRNPG in human myometrial cells, ER delta 7 is also knocked down. However, ER alpha 66, which is a full-length ER alpha isoform, stays the same. These findings further demonstrate that hnRNPG regulates alternative splicing of ER delta 7. Now let's look at the relationship between MET-L3 and hnRNPG. As I mentioned, hnRNPG is a known reader of M6A. MET-L3, on the other hand, is a writer of M6A, meaning that it places the M6A marks on RNA. After performing the Western blot, we see that MET-L3 expression oscillates during mice gestation. Between days 14 and 17, we see an increase in expression and a substantial decrease by day 18, a considerable increase by day 19, and an absence in expression during labor. On the other hand, with hnRNPG, we see that its expression is more or less the highest between days 14 and 17 of gestation. However, between days 18 and 19, we see a pronounced decrease in its levels. Based on this diagram, we see that there's no direct relationship between MET-L3 and hnRNPG. This is because MET-L3 has other functions besides writing M6A. For instance, it plays a major role in cap-independent translation. So it may not be the best check for M6A expression during gestation. If in the future we find that other writers in the writer complex, including MET-L14 and WTAP, have no other function except writing M6A, it will be better checks to determine M6A expression during gestation. Next, we added tunicamycin to human myometrial hTERT cells. Tunicamycin stresses the cells, so we added it to determine if it had any expression, had any effect on the expression of MET-L3 and hnRNPG. In the diagram, we can see that under ER stress, so when tunicamycin is added, MET-L3 expression and hnRNPG expression are both upregulated, meaning that they both cause an increase in M6A methylation. Now I'm going to hand it off to Laila, who will talk about the RNA extraction protocol and what we did for a majority of our internship.
So before I go into the protocol, I'm just going to talk about some of the materials that we use. So when I'm speaking about them while I'm presenting on the protocol, you kind of know what it is. So first we have the RNeasy mini kit, which is where we get all of our buffers for the washing and our different tubes. Then we have our centrifuge, which is something we use a lot during this process, which pulls down a lot of our samples that we need. Then we also have the nano drop machine, something that we use to measure the concentrations of our samples. We have our gel, which is kind of like in the middle, and that's just what it looks like when it's loaded. We have the gel documentation machine, which is how we look at our gels once they have ran. And then we also have some examples of a spreadsheet that we developed, so we know how much of the sample and of water that we're putting into the well when we are loading the gel. So what we performed was an RNA extraction protocol using RNeasy mini kit. Approximately 30 milligrams of our animal tissue, which in this case came from the mouse uterus gestational series, was homogenized in RLT buffer with a K-trope. The RNA was bound to a column and then washed multiple times and eluted with RNase-free water. So after this whole process of the RNA extraction, to figure out the concentration of each sample, we used the machine I was just talking about called a nanodrop. And this basically tells us the exact concentration for each sample, only using 1 microliter of the sample. So once we figure out the concentration that we have for each sample, we are going to develop a spreadsheet so that we can figure out how much of the RNA sample we're going to be putting into our agar shell, and also figure out how much water we're going to be putting in as well. So for our agar shell, once this is complete, we run the RNA samples on an agar shell to determine the quality of the RNA. And the quality of the RNA depends on the 28S and the 18S bands seen on the agar shell. So now we're going to talk about seeing the bands. So we extracted RNA from 3 different series of mouse uterus gestation tissue. To quantify the RNA, we measured the optical density, or the OD, at the wavelength of 260 nanometers. So we found on some of our extractions that the RNA was degraded and both the 28S and the 18S bands were not present. As you can see here, the gels are very streaky, and what we want is to kind of see the bands. So we used a E11 as a control because we knew in a previous lab that we did that it was a high quality sample of RNA from a previous RNA extraction that was run. And so we knew that if we loaded, so we allocated the E11 from the previous lab so that we could just use it so it would be ready to load into the wells, and we wouldn't have to go through the whole process of extracting that RNA over and over again. And so we knew that if the E11 became streaky, that indicated that something was wrong with the loading process, like you see in the first gel. But if we loaded the E11 and it came out fine, like the second gel, you can see there's 2 separate bands, but the rest of the wells are streaky. We know that there's something wrong with the washing, or something probably wrong with just the whole RNA extraction that we did. And so we did have some trials and errors in the beginning, but practice makes perfect. And so we proceeded with the next extraction, and we did so with a lot of caution. We literally banned everybody from our workspace, wouldn't let anybody come in. It was on lockdown. We made sure we sprayed and we RNased literally everything, so nothing would have RNase on it, our workspace, our instruments, or our solutions, and made sure they were all RNase free. And so then the next 3 extractions showed up like this. Wow. Some really beautiful bands. You can clearly see everything is separated, the 28S and the 18S, and this proved that these samples had high quality RNA. Here.
To detect changes in epitranscriptomic marking of M6A in mRNA methylation during mice gestation, we use the M6A RNA methylation quantification colorimetric kit to run an ELISA. Before we even ran the ELISA, we needed to have good undegraded RNA samples. So that's why we ran the RNA extraction protocol. So to begin the ELISA procedure, we start with binding the RNA we extracted from the mice to the bottom of these strip wells, as you can see in the second image, using a binding solution. We then use capture and detection antibodies to detect M6A, and then use an enhancer to magnify that detected signal. In order to colorimetrically quantify the absorbance, we add a developer solution to the assay wells so we see a blue color development, and then a stop solution to stop the enzyme reaction. So once the solution turns from a blue color to a yellow color, we can then read the absorbance by using a spectrometer at a wavelength of 450 nanometers. Throughout the procedure, it is important that we wash the assay wells multiple times to wash off any unbound RNA, any remaining capture and detection antibody. The ELISA protocol allowed us to measure the M6A gestational changes, quantified to nanogram quantities using a diluted positive control of a known M6A concentration. So these are the results that we got after performing our ELISA. Using the linear regression graph we created from our data, we created a bar graph relating M6A RNA concentration to each gestational day. The optical density from the linear regression graph was changed to nanograms of M6A RNA because the concentration of M6A is proportional to the optical density that was measured. From our data, we can see that M6A concentration is highest on day 15. We also see that the concentration at day 15 is almost double that of the concentration at day 13. However, the M6A concentration then drops at day 17 and stays pretty much that way till term labor. Originally, we hypothesized that we would see the largest increases in M6A concentration during the days where the uterus stretches the fastest. This data supports our original hypothesis because as the uterus quickly grows from days 13 to 15, as we showed in like the intro slides, M6A concentration correspondingly increases. In conclusion, our findings implicate that M6A is gestationally regulated. From the ELISA results that we see that M6A concentration is highest at day 15, which correlates to the point where the mice are growing at their fastest. We also found out that when we added tunicamycin to human uterine cells, both MET-L3 and hnRNPG expression are upregulated. From this, we propose that ER stress regulates M6A. In the future, just as we did with mouse uterine tissue samples, Dr. Surya is planning on performing RNA extractions and the ELISA procedures with human uterine tissue samples to eventually perform a Myocyte. The Myocyte is an immunoprecipitation using antibodies and RNA fragments that are later cloned to be sequenced. Using the Myocyte instrument, Dr. Surya is planning on sequencing the things that are attached to the M6As. Like I said before, MET-L3 has other functions besides writing M6A. So it may not be the best check for M6A. So Dr. Surya will also study other writers in the writer complex, including MET-L14 and WTAP, to see if their only function they have is writing M6A. If it is, they will be better checks than MET-L3 to determine patterns in M6A expression during gestation. Before we end our presentation, we would like to take a minute and thank some of the people here today. To begin, we would like to thank Dr. Surya, Dr. Condon, and Francis for teaching us so much during our internship these last 8 weeks. Not only did we learn a lot regarding research, but we also learned a great deal about a variety of lab protocols, the importance of having uncontaminated samples, how to use certain instruments, and how to analyze data, and much, much more. Most importantly, however, our mentors showed us that it was okay to make mistakes and have failures. Like Laila mentioned, our experience with the RNA extractions showed us that we shouldn't give up because negative data is still data. It was because of that data that challenged us to think about what went wrong in our procedure and allowed us to make changes before we ran the experiment again. It was after running the experiment multiple times that we eventually got satisfactory results. Next, we'd like to thank all the mentors that spoke to us during our Wednesday lectures. We really appreciate all the advice you gave us regarding our future careers. Next, we'd like to thank ELISA for organizing all the fun activities during our relationship and internship. Oh my goodness.
It's okay, you have a relationship.
During our internship. She made sure to take lots and lots of pictures, so thank you for that, Alyssa. We will never forget the amazing time we had here. Finally, we would like to thank Dr. Moore for starting this program at Wayne and for giving us the opportunity to partake in such an amazing experience. And that's it with our presentation. Thank you for your time, and we hope you enjoyed it.
I'm sure they not have questions for you. They have questions? You're very kind with you. Very good. Let me